Hey everybody, my name's David Wolking. I'm a Jim Ellis awardee, fellow, if you will, for 2008. And I also work for the GL-CRSP, the Global Livestock Collaborative Research Support Program at University of California, Davis, where I'm a master's student in international agricultural development. Now, the cool thing about being a Jim Ellis awardee and fellow is that I am currently in Tanzania, studying, as you saw from my amazing title, the NERD Project. Essentially what we're doing is trying to look at an epidemiological study of risk factors associated with diarrheal disease in young and naive livestock populations with pastoralist groups that the GL-CRSP project, called HALI, Health for Animals and Livelihood Improvement, has been engaged with for about 2.5 years. Now a word from our sponsors. The Global Livestock Collaborative Research Support Program is a multidisciplinary research and capacity program active in Central and East Asia and West and East Africa. The GL-CRSP is managed by a management entity based at the University of California, Davis. But GL-CRSP projects are conducted by multiple U.S. universities like UC Davis, Texas A&M, University of Wyoming, and Utah State. It is funded by USAID from the American people.
Fantastic. Now let's get started. Who was Jim Ellis and why are we fellows? Jim Ellis was a systems ecologist and professor at Colorado State University. He was a pioneer in developing a paradigm of humans, herbivores, and rangeland systems, and revolutionized the view of pastoralists as pariahs, rather developing the portrait of pastoralists as opportunistic rangeland resource users, whose traditional, extensive grazing systems serve to integrate with and even benefit the complex dynamics of arid ecosystems. Dr. Ellis's revolutionary work on rangeland systems dynamics and spatial analysis of pastoral production systems has contributed to drastic changes in both rangeland management policy and development projects. In 1998, as principal investigator for the GL-CRSP policy project, Dr. Ellis gave a keynote address to the CRSP biannual conference entitled Extensive Grazing Systems, Persistence Under Political Stress and Environmental Risk. In it, Dr. Ellis questioned approaches to livestock development, seeking to eliminate the mobility and spatial extensiveness that provides the risk-mitigating potential to survive extreme events. His hypothesis, that a major failure of development policy lies in the SIP model, sedentarization leading to intensification, leading to increased production, is wholly inappropriate for dryland ecosystems. Instead, Dr. Ellis argued for a new approach to development, one that considers the economic and ecological constraints facing dryland grazing systems and the critical need to exploit large spatial gradients to maximize risk management for pastoral producers. This theory has been fundamental in developing the current GL-CRSP program. In the words of GL-CRSP Director Tag Dement, Jim's scientific vision was broad and integrated and brought a perspective to rangeland systems that made a major contribution to the understanding of pastoral systems. His work was critical to the development community in a number of ways. First, it provided logical reasons for the failures of past livestock development projects. Second, his work demonstrated the role of research in the development process. And third, his work clearly illustrated the role of spatial scale in unpredictable environments. Well said, Tag. Jim tragically perished several years ago, and in remembrance, the GL-CRSP established the Jim Ellis Mentorship Program for graduate students.
Fast forward to 2008, and I'm a proud Jim Ellis Fellow. I'm also a master's student in the UC Davis International Agricultural Development Program, lovingly called IAD. IAD is nice. It allows for a broad interdisciplinary approach to a research-focused curriculum. As an example, I entered with a concentration on agriculture and resource economics, switched to soil science and nitrogen subsurface modeling, then to water quality, and finally on to epidemiology. It's been a long 2 years, but I've learned a lot. Like when I'm sampling calves and staring down at an iron oxide-rich topsoil, heavily leached and highly acidic, or when I see a borehole merely a kilometer away from another borehole, and reflect on the relative lack of water infrastructure planning in this arid ecosystem. So what am I doing in Tanzania anyway? I'm working with the GL-CRSP Health for Animal and Human Livelihoods project or HALI. It's a collaboration between the Wildlife Health Center at UC Davis, the Sokoine University of Agriculture in Tanzania, and the Wildlife Conservation Society's Ruaha Landscape Program in Tanzania. Essentially, what HALI is investigating is the interface between wildlife, livestock, and human livelihoods in pastoral communities near the Ruaha National Park and wildlife management areas. You see, HALI's found TB in both livestock and water buffalo, and when mixed in with HIV in the human population, a potential disaster could unfold. It's really a disease surveillance project, sampling wildlife and livestock and running a series of surveys on pastoral households to assess the potential for improving rural livelihoods in the area. After consulting with the HALI team, we came up with a study to complement the HALI disease surveillance data. HALI's been sampling livestock in the area for TB and other pathogens, including Cryptosporidium parvum and Giardia. They haven't really found much in terms of crypto and Giardia, but they haven't been sampling naive livestock populations. You see, crypto is pretty nasty. It's a protozoal pathogen transmitted primarily through a fecal-oral route, but it can also infect through the ingestion of a contaminated water supply. It gets in your intestines. It burrows deep into the tissue, and when it's ready, it stimulates a reaction in your gut to flush. In the diarrhea are oocysts, or spores of crypto. They're free, and they look for another host to infect. Crypto is a major cause of morbidity and mortality in livestock populations, and also in immunosuppressed human populations. It inflicts calves and babies who aren't really ready to handle the germs of planet Earth, and also those whose bodies are ready to succumb to them, the elderly, those with HIV. We want to know if there's crypto in livestock in Ruaha, in the HALI pastoral communities. We want to know if it's a threat, and if it is, we also want to know what factors increase or decrease this threat. Factors like management practices, herd composition. So I created NERD, the Neonate Research on Ruaha Diarrhea Project, and Jim Ellis was kind enough to call me his fellow. I'm in Ruaha now. I've been here for about a month. The study's up and running. I've just received preliminary research approval from the Tanzania Commission on Science and Technology, so things are underway. We're implementing a survey, questions about births in the herd, disease in the herd, how disease is diagnosed, how disease is treated and prevented, if diarrhea is a problem, if water is shared, how manure is managed, lots of questions on poo and feces. And we're sampling feces in calves. We put on a glove, we grab a calf, and we delicately insert a finger into its anal cavity. Then you preserve it in formalin and send it to the lab. I'm working with the Sokoine University Faculty of Veterinary Medicine. There, the fecal samples are analyzed through a fluorescence microscope. The sample is stained, so any crypto or giardiosis and cysts light up, kind of a lime green, like lights on a Christmas tree. We count them and establish prevalence of infection. Then, we'll compare the prevalence, percentage of infected animals, with the survey data to come up with some factors that may explain the infection, or at least correlate with it. We're also going to go to some households that are weird. Households that do things differently. Good things and bad things. We're going to watch them for a full day to make sure the behavior is relatively consistent with the survey results. So far, we've surveyed about 24 households and sampled about 131 animals. Got a great field assistant named Asha from Malinzanga, one of our project villages, and she's made life pretty easy. Some of our hypotheses are slowly being validated as well. For example, sharing water sources in the household may contribute to an increased risk of fecal oil transmission, as cow or goat muzzles that poke around in manure all day long poke also into your water supply, maybe in a bucket, maybe in a basin used to wash your clothes. I think you see what I mean here. Goats love bucket water. They're little thieves. They also may be huge environmental pathogen emiters, both for crypto and Giardia. Next time, I'll share some preliminary results from the survey, a halftime show of sorts. I'll introduce you to Asha, and also my girlfriend Misty, who's coming to help out on the 19th of September. She's a fantastic photographer, so I should have some real nice photos to share with you. Thanks for putting up with me. Thanks to Susan and Tag at the GL-CRSP, and Deanna, Jana, and Harrison of the HALI project. And of course, thanks to Jim Ellis.